Next up is Dr. Lipika Goyal from Massachusetts General Hospital, who will be speaking on the topic of response and resistance to FGFR inhibitor therapy.
Thank you, Peter. That was excellent and really set up a lot of what I was going to say. And I was happy that you said BGJ later on in your talk, so I didn't have to say infigratinib. That's right. I think we should call it Infi maybe, but we'll stick with BGJ. So 2017, first I wanted to say thank you to Dr. Bardisi and to Dr. Kelly and to Stacy and the Cholangiocarcinoma Foundation for giving me the privilege of talking about this exciting topic here at this awesome conference. 2017 has been a landmark year for FGFR positive cholangio. And it's mainly because there's so many drugs that are showing promise, and there's so many companies that have committed to do trials in cholangiocarcinoma. And why? Why are there so many companies that are committing to a rare subtype of a rare disease? Because the drugs are working. It's one of the few times that I actually call my patients, I disturb them on the weekend, is when I see they have an FGFR2 fusion. And I call them and I say, guess what? I'm really excited you have this fusion. We have great drugs for you. But it's also a really good time to be talking about resistance, because while people are having tremendous tumor shrinkage in some cases and on these drugs for several months, one, they don't work for everybody, as Dr. Hammerman alluded to, and sometimes people are only responding maybe 10 or 20% and only for 4 to 6 months. So we need to understand why. So this, Dr. Hammerman went over in detail, Dr. Javale and Dr. Saab's phase 2 study of BGJ. But 2017 has also seen a panoply of other FGFR inhibitors that are showing promise in patients with cholangiocarcinoma and showing a significant degree of shrinkage. How does our disease compare to other oncogene-addicted tumors? So when you look at ALK-positive lung cancer, crizotinib, the response rate is more around 60 to 70%, and the progression-free survival is around 9 to 10 months. So why is it that with BGJ we're seeing not as robust a response? There are many reasons, but one of them could be resistance. And so there's intrinsic, adaptive, and acquired resistance. This is relatively simplistic, but one of the ways I think about it is the patients on the left, they are not really responding at all, and they might have some sort of intrinsic resistance, like a co-mutation that's not letting them respond to the FGFR inhibitor, or maybe the way the fusion is formed, they're losing their kinase domain and the drug cannot bind. Adaptive resistance, I think of that as people are initially responding, but then there's some quick compensatory mechanism that leads to signaling that, again, takes away their response to the FGFR inhibitor. And then acquired resistance, I think of it as people who initially did respond and potentially responded for 6 to 8 months, but then they developed some sort of mechanism that made them stop responding. And that could be a second site mutation in the FGFR2 kinase domain. That could be upregulation of bypass pathways. And there are FGFR-dependent and FGFR-independent mechanisms, and I'm going to focus on acquired resistance and specifically FGFR-dependent mechanisms that we're discovering in the clinic. So I'll just share one slide about our previous work on acquired resistance to BGJ. Then I'll go directly into how we are overcoming resistance to prior FGFR inhibitor resistance with this irreversible inhibitor, TAS-120. And then finally, I'll end with some cases in the clinic of people who got TAS-120 and then developed clinical resistance. So this is work that was done with Dr. Saha at the University of Washington, with Dr. Bardisi, with Dr. Zhu, several people in this room. We basically looked at 3 patients that we enrolled on the multi-center phase 2 trial of BGJ. All of them had FGFR2 fusions. All of them had cholangiocarcinoma. They all got BGJ, and because we knew from previous studies that these patients were likely to develop resistance, in collaboration with Ryan Corcoran, we did serial blood and tissue biopsy collection, and we found that all 3 patients developed a mutation in the gatekeeper. That's the red one, V564F, and 2 of the patients actually developed 5 different mutations. So they developed polyclonal resistance in the FGFR2 kinase domain. That makes our job very tricky as oncologists, because what are we going to do next when you have to deal with 5 mutations? So Dr. Nathaniel Gray and his team at Dana-Farber, and Dr. Hammerman was part of his team, they worked on developing different drugs that can overcome resistance with these mutations. So they developed some irreversible inhibitors. They didn't actually go into the clinic, but they formed the blueprint for a drug that did go into the clinic, which was TAS-120. And as you can see, TAS-120 is a covalently binding, highly selective drug against all three FGFRs, 1 through 4, with single-digit nanomolar potency. It shows dose-dependent tumor growth inhibition in models with FGFR2 amplified gastric cancer. And then a very important aspect of this drug is the company did some studies and showed that the IC50s for the precise mutations that people developed after BGJ, the IC50s are single-digit nanomolar. So the idea was, our hypothesis was, that if we give TAS-120 to the patients after they develop resistance to other FGFR inhibitors, potentially they'll get some benefit. So we had 4 shots on goal. We had 4 patients who came to us who got a previous FGFR inhibitor and then were looking for next drugs. It was a little bit intentional to have these 4 burly men because they're kind of like FGFR2 mutations in the way of TAS-120. So this is the first patient. So all the slides are going to be set up the same. In the top left corner is going to be a little bit of clinical information about the patient. Top right corner is going to be the ctDNA analysis. And the bottom is going to be the radiology. So this first patient was a 63-year-old Caucasian female. She is patient 1 from the paper who had a ZMYM4 fusion partner. And she had BGJ for 5.6 months, and she developed 5 different mutations in her circulating tumor DNA analysis. When she went on to TAS-120, 3 of these mutations became undetectable and eventually started to grow again, or started to arise again, 2 of them. But 2 of them, the gatekeeper mutation and 659M, those 2 didn't go down significantly. And this was interesting because TAS-120 was supposed to inhibit them, but something that we learned about in the clinic and are studying in further detail with Dr. Bardisi. So she had 7.4 months of stable disease, so she certainly got clinical benefit, but she did not have shrinkage. The second patient was patient number 2, 73-year-old woman who had also an FGFR2 fusion. She is the one of 4 patients on BGJ who did not develop a gatekeeper mutation. She developed 2 other mutations that were shown to predict for resistance to FGFR inhibitors in preclinical models. And when she got TAS-120, these 2 mutations became undetectable. And she had a nearly 70% response on BGJ, and then she had a 76% response on TAS-120. And she is still on trial, and she's been on TAS-120 for 15 months. So this has been a remarkable response for this patient after resistance. The next patient is a patient with an NRAP fusion partner, and he was on BGJ for 7 months. Then in between, he got pembrolizumab and FOLFOX, and when he came to us at MGH, we did a baseline circulating tumor DNA analysis and didn't find any mutations, but he also had a 45% response when he went on to TAS-120. And finally, the 4th patient is a patient who had a great response to DEBIO-1347, another FGFR inhibitor. He had a 46% response, was on it for 11.2 months, and got empiric chemotherapy in between. And then just before he started TAS-120, we did a biopsy, and we found that he had 3 different mutations. And that last mutation we had not seen on BGJ. He went on to TAS-120, and he's had a 22% response at 6 weeks, and he's continuing to be on it right now. So this is the summary of those 4 patients. As you can see, let me see if this pointer works. Yep. All of them had a FGFR2 fusion. 3 of them went on BGJ, 1 on to Debio. All of them had a response on the first inhibitor. All of them were on for 5 to 12 months. 3 out of 4 of them developed mutations, either in circulating tumor DNA analysis or on the biopsy. And then they all had clinical benefit. 3 of them had tumor shrinkage, but 1 of them had stable disease. But there's still several questions about these patients. So number 1, with the first patient who had stable disease, why did she not have shrinkage? What is TAS-120 doing that, is it truly not able to inhibit these mutations properly, and that's why she didn't have it? Is it because TAS-120 can inhibit 1 mutation at a time, but if there are multiple mutations, it has difficulty doing that? Are these mutations in cis with each other and in cis with the fusion? Are they in the same cell or are they in different cells? Does it make a difference what fusion partner these patients have? Because the patient with the SORBS1 fusion, she was very sensitive to both inhibitors. So is that playing a role in some way? And then, as we were saying with the patient on Debio, he had a new mutation that we hadn't seen on BGJ. So do different inhibitors lead to different kinds of resistance mutations? And then the patient who had the NRAP fusion, he had intervening chemotherapy. So was it that he just regrew his sensitive clones and those are the ones that are responding, or did he actually have some sort of resistance mechanism that we could not detect on circulating tumor DNA or on biopsy? So next we'll go to acquired resistance on TAS120, and we'll talk about 3 patients who had resistance. One of the most interesting is the first patient who had a BIC1 fusion, which is the most common fusion you see with FGFR. And she had a mutation in the cysteine residue that this irreversible inhibitor actually binds to. And this is some interesting work that Dr. Nathaniel Gray has also looked into. This has been predicted. We see this also with osimertinib, which is the irreversible inhibitor for EGFR. You see a C797S mutation. So it's, again, precisely the cysteine residue that these inhibitors bind to. So it's an understandable mutation that she emerged, but she emerged 7 different mutations. Interestingly, when we biopsied her bone lesion, because she was still responding in the liver and she had oligometastatic progression, when we biopsied her bone lesion, she didn't have any of these 7 mutations. So it begs the question, were any of these 7 mutations actually the relevant mutation that was driving her progression in the bones, or was it something else? And to be totally transparent, for all these ctDNA graphs, you often pick up many, many mutations on ctDNA. So for simplicity, I just included the FGFR2 mutations, but many of these patients also had mutations in other genes as well. This is another patient, a patient of Dr. Andrew Zhu's, and she had multiple lung and liver lesions. And a week or 2 into taking TAS120, she walked into Dr. Zhu's office and said, "Dr. Zhu, I think this drug is working," because her cough got significantly better. And she did very well for about 6 months and then started to progress more, and she came off at 8.5 months. And she interestingly developed a gatekeeper mutation. So this was interesting because, again, we're thinking that TAS120 should generally overcome this mutation. So maybe it was a cooperating mutation, we're not sure. And then the last patient is a 57-year-old patient who had a CBX5 fusion partner, and she developed 4 different mutations. So again, this is the summary of the different patients who were on TAS120. All had tumor shrinkage. They were all on it for 7 to 13 months, and they all developed mutations in the FGFR2 kinase domain. So this is these are the IC50s that we should be seeing. So we're doing work with Dr. Bardisi to understand a little bit more about the mechanistic understanding of TAS120. So the summary of our patients is that we had 4 patients on BGJ that progressed, and we were able to identify mechanisms for resistance, potential mechanisms for resistance to all 4. They all need to be validated. And of our TAS patients, we identified resistance mechanisms in 4. So I'll skip this slide in the interest of time because I think Dr. Hammerman already went over it. It's just the idea that we need potentially FGFR2 selective drugs, because when you get the hyperphosphatemia, we sometimes have to do dose reductions and dose interruptions, and that can impact efficacy and the overall resistance patterns that we're seeing. So this is my second to last slide, and this is about why I have hope. And this is the story in lung cancer. So the ALK fusion was identified in lung cancer in 2007, and the EGFR mutation was identified in lung cancer in 2004. And now, 10 to 15 years later, there are 4 to 5 drugs that are approved in each. So the question for us is, what can we accomplish in the next 5 years? Because our clock started ticking in 2013, because that's when FGFR2 fusions were identified in cholangio. So in the next 5 years, I hope that we are able to approve a couple of drugs for our patients. And I think our hill is a little steeper to climb because of all the reasons that Dr. Hammerman alluded to in terms of why FGFR is a little bit different than some of the other oncogenes. But I think we're standing on the shoulders of giants because we now understand a lot more about getting serial biopsies and serial DNA, and we're much better with genomics than we were 10 years ago. So my final slide is, instead of doing a summary, I decided to do a call to action. So I love this cartoon because we all know that tumors evolve, and tumors from 1 year ago are not what tumors look like now. And so for physicians, I was talking to one of my colleagues over at Gustave Roussy in France, and we were talking about how the #1 barrier to getting serial biopsies and blood is actually the fear of the physician just asking the patient, you know? So as we move into more clinical trials, please do ask. Our patients are incredibly generous. Please be meticulous about providing high-fidelity data to the pharma companies about the FGFR alterations and the co-mutations. In the great BGJ paper that came out, understanding a little bit more about specifically what were the co-mutations that patients had and how were they responding specifically to BGJ, where they were on the waterfall plot, can help our scientists give us a little bit more understanding about what could be happening on a biological level. To pharma, thank you for committing to our patients, and please write in serial blood and optional biopsies, which many of the companies are already doing, because some institutions don't have the ability to do this as a research protocol, so to have that as an option on protocols makes a big difference to be able to serially collect this kind of information. To our scientists, please point us in the direction of where we need to go, because we can certainly do clinical trials, but the more informed we are by what the biology is, the better we have a chance of these drugs working in terms of what combinations should we be using. And to our patients in finale, just thank you for having the courage to go onto clinical trials. Thank you for your grace in coming oftentimes hours by car and by plane to come to us in rain and snow. And thank you for spreading the word. A lot of my patients feel that there is a tremendous community out there because of everything you do in terms of blogging and being willing to talk to people. So I am hopeful that with all your hard work and all of our hard work, we're going to continue to make a dent in this disease. Thank you.